Hello students. So today our chapter is chromosomal theory of inheritance. Before this we completed the dihybrid cross, right? This theory, one chromosomal theory of inheritance. This theory is we can say proposed by who proposed this theory? It is Walter Sutton and we can say another Theodore Barberi. This theory it is proposed by Walter Sutton and we can say Walter Sutton and second one is Theodore Barberi, right? Barberi, right? So they proposed this theory, chromosomal theory of inheritance, because they first saw in cell division, they saw these chromosomes, they get separate and move toward the underside in anaphase condition, right? Then they propose this theory, chromosomal theory of inheritance, right? One thing is there, and in which second point is there, in which year this theory is proposed. So they proposed this theory in 1902, right? So you understand, chromosomal theory of inheritance means through chromosomes he gives how a genes, or we can say traits, they transfer from one parent to another parent, right? Now what is given in your NCERT, we will read. Mendel published his work on inheritance of characters in 1865. Already we know in 1865 Mendel published his work, right? But for several reasons it remained unrecognized till 1900, right? Firstly, means in his life he cannot understand, he cannot he never think that he become the father of genetics, right? Never he can. Firstly, what are the reason why it is remained unrecognized? First is there, because communication was not easy as it is now. In those days, what we know, where here this time information technology is very high, right? So we can say, but at that time communication was not easy. No phone, no Android phone. We can, no one understand that time. We say this idea is not in their mind that sometime will come in at which we can talk to each other with the phone or Android phone. Is no one understand that time. That time only paper is there, and paper is not available for every person. That's why here communication was not easy at that time. First, second reason, what is here? That's what could not be widely published. First thing, because communication is not easy in those days, and his work is also could not be widely published. It is published only in its known city, not any other places, right? Secondly, his concept of gene at the time, Mendel said these genes as a factor, right? So according to Mendel, factor in Mendel's word are as stable. Mendel said they are stable. They are stable and discrete units. They are stable and discrete units, right? Discrete units that controlled the expression of trait. First he said they are stable and discrete units, but he cannot give any proof of the presence of genes or factors, and of the pair of alleles which did not blend. That is another, did not blend. He said they did not blend with each other, and at that time biologists they did not accept this one, because many variations are present in nature. So variation means what? They think these characters they are blend with each other and form a variation. Simply that time they think these characters, these traits, they blend with each other and a new variation is found, intermediate, right? But Mendel say they did not blend with each other, was not accepted by his contemporaries, right, as an explanation for apparently continuous variation seen in the nature. Because they see continuous variation, they already see in the nature. Variation means they simply know that these are the due to the blending of factors or genes. But Mendel said they are not blend each other. That was not acceptable at that time. Third means what? Mendel approach of using mathematics. Mathematics means simply I said p square plus 2pq plus q square equals to 1. It is hardly been verification, but Mendel also said then 1:2:1 ratio is there. 1 means what? 1 square means 1. That is capital T, small t, and capital T, small t. So because Mendel used mathematics, statics to explain the biological phenomenon, it was totally new and unacceptable. It is totally new and unacceptable to many of the biologists at his time. These three reasons are there. First, what is given here? Because communication was not. Second, what is there? He said genes are responsible for, and in Mendel word it is factors. Factors are responsible, and these factors are stable and discrete units, and they did not blend each other. That was not acceptable. Third, he used mathematics in biology. That was also not acceptable. It is even totally new and unacceptable to many of the biologists of his time. Right? Finally, though Mendel's work was suggested that the factor genes were discrete unit, but he could not provide any physical proof, because at that time the scientists they want only proof, right? But he could not provide any physical proof for the existence of factors or say what they were made up of, because at that time we have no idea about the genes. How could we understand what they were made up of? We have no idea at that time also. So he did not give the proof. That are the reasons. Means here 4 reasons are given. Simply we can say 3 reasons are there, but we can in this passage 4 reasons. Why I am saying 4 reasons? First, communication gap. Second one is he said factors. Factors are stable and discrete units which never blend with each other. That is unacceptable at that time. Third, he used mathematics in biology. It is totally unacceptable by that at that time. Fourth one is also there because he did not give any proof, physical proof for the existence of these factors or genes, right? Or he did not tell them that what they were made up of. These are the reasons why Mendel's failed at that time, right? Mendel is not failed, but he is not become popular, or he is not credited for this discovery. In 1900, after 34 years, 1865 he published his book, right? And in 1900 means up to 34 years he is in dark side. No one knew about his practices or practicals, or we can say about his experiments. But after 34 years, in 1900, 3 scientists. Who scientists? Hugo de Vries, he works on evening primrose. Karl Karons, he works in maize, right? And von Schermark, right? Von Schermark, multiple different plants he used. So de Vries, he worked in primrose. Karl Karons, we can say de Vries on evening primrose. Second is Karl Karons, he worked in maize. And third one is our von Schermark. He worked in different plants, right? They independently rediscovered the Mendel's work. They independently rediscovered the Mendel's result. Independent means in their countries, right? On the inheritance of characters. Also by this time, due to advancement of microscopy, because at that time microscopy is invented, that were taking place, scientists were able to carefully observe the cell division, right? This led to the discovery of structure in the nucleus that appeared to double and divide just before each cell division. When they see the nucleus, they see the chromosomes. Chromosomes means they doubled first, then what happens? In anaphase, they get divided, right? These were called as chromosomes. Chromo means color, and som means bodies. Colored bodies, why? Because after staining, they are colored, they stain. So they are called colored bodies, and that means what? Chromosomes, colored bodies, as they were visualized by staining, right? By 1902, after 1900, these three scientists, they rediscovered the Mendel's experiment, right? In after 2 years, 1902, the chromosome movement during meiosis had been worked out. Meiosis, you know, reduction or division. So the chromosome movement during meiosis, it has been worked out in 1902. So you understand, 1865, Mendel published his experiments in a magazine, right? And in Broun City only, right? Then no one understand this one. In 2000, these three scientists, Miss Hoover, De Vries, Correns, and von Schermer, they rediscovered the Mendel's experiments independently. In 1902, the chromosomal movement during meiosis is also discovered or had been worked out, right? And when they understand about the chromosomes, right? Who worked out? That is two another scientists, and I'll tell you before this one who is there. It is Walter Sutton and Theodore Bavin. Chromosome 1902, the chromosome movement during meiosis had been worked out, right? Walter Sutton and Theodore Baveri noted that the behavior of chromosomes was parallel to the behavior of genes, and they use chromosome movement to explain the Mendel's law, because they use chromosome movement to explain the Mendel's law. That's why they proposed a theory, that is chromosomal theory of inheritance, which is called chromosomal theory of inheritance. Recall that you have studied the behavior of chromosome during the mitosis, means equational division, and during the meiosis, that is called reduction division, in which number of chromosome is reduced by half, right? Important thing to remember are that chromosomes as well as genes occurs in pairs. We know very well, 23. In humans, what happens? 46 chromosomes are there, but they are in pair. 23 pairs of chromosomes are there. Each allele, already we read. Allele means what? Allele means it. Means we can say genes which are present on homologous chromosome at the same loci, same location. Loci means here what? Location. Means genes are also present in pair, right? Chromosome also present in pair. The two alleles of a gene pair are located on homologous site on homologous chromosome. Simply it is given two alleles, not a single allele. Two alleles of a gene pair are located on homologous site on homologous chromosome, right? So homologous site on homologous chromosome, what do we understand this one by? Look here. Suppose that I form two, one chromosome, second chromosome, right? Linear shape of chromosome, because we have linear shape of chromosome. One gene is there and the gene is there. A, B, A. These are two alleles. A, B. One this side, one this side, another this side, another side. So same. That is called homologous site. Same site, right? Homologous site on homologous. These are first pair chromosome. This is also first pair chromosome. So both are the first pairs chromosome, right? And these are genes. We can say genes, because these two, they are called as allele. These two pair, they are called as, this one and this one is called allele, right? One figure is given. When we consider this figure, we understand here cell division stage are given. You can see here G1 phase. G1 phase means growth first phase. That is what happens here. Here cell enlarged and cytoplasm is there. Cytoplasmic growth is there. In G2 phase, what happened? This one is G2. Before G2, S phase is there. Here S phase. S phase means what? Here replication. replication or duplication may be same of DNA happens. Now replication means what happens? Each chromosome, they have two sister chromatids, one and second, A and B. You can see here 1, 2 chromosome and these 4 chromosomes are there. That meiosis first. Now G2 phase means some other proteins are formed. This is growth second phase. So between this G1 and G2 S phase is there where this single chromosome, single chromatids. Now here what happens? Single chromosome but 2 chromatids are formed because here number of chromatids are 4n. Chromatids means what? DNA content, right? But chromosome number is 2n. Do you understand here? Chromosomes 2, 1, 2, 3, 4 is there. 1, 2, 3, 4 is there. Number of chromatin thread is increased. Here what happens? Meiosis first, anaphase. Anaphase means what? They get separate from one another. Right? Now here, this is the spindle fiber, this side is spindle fiber. Now they are separation. So meiosis first, anaphase, what happens? Meiosis second, anaphase is given here. Meiosis first means they separate from that side and that separate, this separate to this one side. We can say here, in meiosis first, these chromosome, they comes to that place. Right? This come to that place. And this chromosome, it come to this place. This one to that place, this one to that place. Right? You understand? They come to different protein. So here what happens? N chromosome to this side and chromosome to that side. This is 2n chromosomes. Now here N to this side and to this side. Green and this pink color, this goes to lower side and this yellow and red to upper side. And meiosis second means what happens? Now these two, they once more separate. Here these are separation. And this, now here 4 germ cells are formed. These are germ cells, or we can say daughter cells. 4 daughter cells are formed. Do you understand this one? Suppose that, here you understand, suppose that this one is a sperm cell. This one is sperm mother cell. Spermatogonia is, I am asking about spermatogonia. What happens? Sperm cells. Right? Spermatogonia, sperm mother cells. Right? Then what happens? Meiosis first. Meiosis first means here 2 types of cells are formed. This one, it contains 22 plus X. It contains 22 plus Y. Now you understand this one is also 22 plus Y, 22 plus Y, 22 plus X, 22 plus X. That type is there. Do you understand? 2 different type, both are same. These both are same. X, X, these are both are same. Means Y, Y. We understand this one. So this figure, it explains us how a meiosis means how chromosomal movement occurs in meiosis. Simply. Okay. Now a table is given here. A comparison between the behavior of chromosomes. A table is given in between the behavior of chromosome and genes. Now 2 tables are given, A and B, and one question is also given here. Sometimes you get confused when you see column A, column B. It occurs in pair, both are same. Both statements are same. Means we cannot understand which one is gene and which one is chromosome. Right? Second, segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. Segregate at the time of gamete formation. They segregate at the time of gamete formation in meiosis, right? Such that only one of each pair is transmitted to a gamete. Okay, correct. Segregate at gamete formation and only one of each pair is transmitted to a gamete. Both are same. We can say. Means we cannot understand here also. So this second, we cannot understand which one is chromosome or which one is gene, because both statements are same. Only one of each pair is transmitted to a gamete, That means we cannot understand which one is right. Both statements are also same. Segregate at the time of gamete formation. It is also called segregate at the time at gamete formation or at the time of gamete formation. Means both statements are same. That means through these 2 statements, we cannot define which one is chromosome or which one is genes. Okay. Now here we have only one third column is there. Independent pairs segregate independently of each other. independent pairs segregate independently of each other, right? As for example here, 2, we can say 1 and 2. These are chromosomes, right? These are chromosomes. A pair of chromosomes. When in anaphase, what happens? They independent pairs segregate independently of each other. So these are independent pairs. They segregate. One move to that side, one move to that side, right? In a nucleus, independently of each other. They are not means independent of any one of them. So they move independently. Means here these are chromosomes who moves independently. Okay. Here what given? One pair segregates independently of another pair. Here pair is given. One pair is segregated independently. And now look here. Suppose that I forms it 2 chromosomes. Here 1, 2. A, B gene is there. And here also A and B genes are there. So this pair segregate, right? What is given? One pair. So this we can say A and B, these pairs. These pair segregates independently of another pair. So this pair means A and A, they segregates. They can move to this side or this side, we can say, right? Segregates each other independently to other pair. This one is another pair. Do you understand what I am saying? Look here. This one is another pair. Each pair segregates independently to another pair. This is pair. We can say these are pair. When we say each pair segregates independently to another pair, when we are talking about the pairs, that means we are not talking about the chromosomes, because chromosomes are pairs are not segregated. Here we say chromosomes are segregated, right? From one pair, one chromosome segregated to one side, another chromosome segregated to another side, right? Here what is given here? One pair, this is the pair, segregates from the another pair. That means what? It is, we are talking about the genes. One pair of gene, we can say one pair of gene segregates independently another pair of gene. Do you understand? Now we can correct here, we can define here that B means what? Gene. We are talking about the gene, and A means what? It is chromosomes, right? Here this question is given. Can you tell which of these column A or B represents the chromosome and which represents the gene? So we can understand that A represents the chromosome, because here independent pairs segregate independently of each other. That means this each other, they segregate each other from, right? But here what is given? One pair segregates independently of another pair. Here one pair means this pair, it segregates independently from another pair to BB, right? I think you understand which one is gene and which one is chromosomes. How did you decide? By this 3 statement. So this third column we can decide. We can be decide, because here pair segregates, but here single chromosome segregates independently of each other. Okay. Now here, during anaphase of meiosis first, the 2 chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of 4 different color, the left and the right column. In the left column, possibility first is given, that is here, right? And that is orange, and green is segregating together. But in the right hand here, right, possibility second, that is orange chromosome is segregated with the red chromosome. Two possibilities given here, right? And we can see here what possibilities are there. Possibility first and possibility second. Here, in this color body, we can see this one is orange and this one is orange and red. Two things are given. Long orange and short green chromosome. Long orange and short green chromosome, right? And long yellow and short red chromosome. This one is long yellow and this one is short red chromosome. Okay, when they segregate in meiosis second anaphase, it is meiosis first anaphase. Meiosis first anaphase, what happens? chromosome reduced, number of chromosome is reduced, means here what happens, 23 chromosome moves to one pole and 23 chromosome they moves to another pole, right? In anaphase first. Anaphase second means what happens, here chromatin threads they get separated, right? Kinetochore gets separated. Here what happens, in meiosis first, these chromosomes they get segregated. Meiosis first means what happens, they get segregated. Here you also, here what happens, with orange red is given and with yellow green is given. Here what happens, with yellow red is given, orange green is given, just opposite. Here just opposite. Meiosis second means a single first daughter cell, this, right? It contains orange and green, and second daughter cell it contains yellow and red. But here what happens, look, this it contains orange and red, this contains orange and green, means here, right? Now germ cells means what happens, these two, here you understand, these two it forms orange and green, orange and green, orange and red, orange and red, 4 daughter cells are formed. But here what happens, this orange and red, orange and red, and here orange and green and orange and green. Independent assortment of chromosomes, simply I tell you here, you can understand. In case of independent assortment, what happens, look here, for linkage. And what is the difference between linkage and independent assortment? Simply in your mind it comes, what is the difference between linkage and independent assortment? Is there any difference or they both are same? So when we consider the linkage, so linkage means what happens, here suppose that we have 2 chromosomes, they are homologous chromosomes. We are talking about only, here we are talking about only homologous chromosomes, right? First pair of male and first pair of female. Here we have two, suppose that 2 genes are there, capital A and capital B. This side only A, small a and small b. That is called linkage. And here we get only 2 types of gametes. First is our capital A, capital B, right? This gamete, or second means we have A and B. This gamete, 2 types. But in case of crossing over, suppose that this 2 is there, but in crossing over what happens? Exchange of genes. Then what we have? Capital A, small a, capital B. So this, through this one, right? Through this one we get 4 types of gametes, that is capital A, capital B. Second one is, it is our capital A, small b. Third is our small a, capital B, and fourth one is our small a, small b. So 4 types of gametes are formed, right? Means 4 types of gametes are formed, right? Here you also see, due to the possibilities, 4 types of gametes are formed. These are different chromosomes, right? We can see here, you can see here 4 types of gametes are formed, 1, 2, 3 and 4, right? So 4 types of gametes are formed in case of crossing over, and in linkage what happens, we have only 2 types of gametes are formed, right? That is the difference between this one. Okay, here simple thing is given here. They first, what happens, number of chromosomes they are reduced, means half number of chromosomes in meiosis first. Meiosis second means what happens, number of chromosomes are not reduced, but these chromatin threads they get separated, right? Now Sutton and Boveri argued that the pairing and the separation of pair of chromosomes would lead to the segregation of a pair of factor they carried. Pair of factor means what? These are genes, right? What is given here? The pairing and the separation of a pair of chromosome would lead to the segregation of a pair of factor they carried. Segregation means what? They separate. Separation of factor. Factor means what? That is genes. Sutton united the knowledge of chromosomal segregation with Mendelian principle and called it the chromosomal theory of inheritance. Actually, Sutton and Boveri both are responsible for the proposal of this chromosomal theory of inheritance, right? But especially when we talking about the special, especially who is there, you understand here in our NCERT, because it is given Sutton, right? This Sutton. united the knowledge of chromosomal segregation with Mendelian principle and called it chromosomal theory of inheritance. Do you understand who proposed? So suppose that a MCQ, if it is given certain separately given, so you have to up the certain only, right? But certain and Baveri given, so it is also correct. Certain and Baveri both are also correct. Now, following this synthesis of ideas, experimental verification of chromosomal theory of inheritance by Thomas Hunt Morgan. He is also called as father of practical genetics. Father of practical genetics, right? Thomas Hunt Morgan and his colleagues lead to discovering on the basis for the variation that sexual reproduction produced. Morgan worked with a teeny fruit fly, that name is Drosophila, right? Drosophila melanogaster. This is a fruit fly, right? Drosophila melanogaster, which were found very suitable for such studies. Why they are very suitable for such studies? Why he used this one? They could be grown on, first thing, they could be grown on simple synthetic medium in the laboratory. They can be grown into the synthetic medium in the laboratory. Easily they can grow. Second, they complete their life cycle in about 2 weeks, mean in 15 days. Yes, life cycle is completed. So lifespan is also less. Lifespan less means what happens? We can study different offsprings, right? And a single mating could produce a large number of progeny flies, right? Also, there was a clear differentiation of the sexes, which one is male or which one is female. The male and female flies are easily distinguishable. Sexual dimorphism is also there. Also, it has many types of heredity variations that can be seen with low power microscope, right? So this is given. So you understand here, Thomas Hunt Morgan means okay, we understand there. Mendel means he used plant means Pisum sativum. Thomas Hunt Morgan, second means De Vries, he used that is evening primrose. Third means what? Coral cornice, he used maize, right? Fourth one is Von Schermer, different plant, right? Then here, Thomas Hunt Morgan, he used Drosophila, animal, first animal. Drosophila melanogaster, which is a tiny fruit fly. Why he used this one? Because it is very suitable for studies. Why it is very suitable for studies? Because it can grow in synthetic medium. First thing is there. Second, its lifespan is very small, means only 15 days or 2 weeks. Third, it can produce more progeny through a single mating, right? Fourth, it has many variations which can be seen by low microscope. Many variations are also there, right? Male and female, they are also shows sexual dimorphism. So these are the reasons why Thomas Hunt Morgan choose this Drosophila flies, melanogaster. Here you understand A and B. A means what? Male is small and this one is female, right? Which one is large, right? And other differences are also there. Red eyes, white eyes. Okay. Now linkage and recombination here. Linkage and recombination. Before going to read this point, I think first we have to read about some terms about the linkage. What is linkage and how it is affected by other factors. Okay. Here, now first question is here, what is linkage? Linkage means what? One thing is, linkage was first seen by Bateson, right? That is, we can say Bateson and Punnett. It is first seen by Bateson and punnet. First seen by, right? But they did not give the name linkage. Only they gave coupling and repulsion phenomenon. So through this one, this Bateson and Punnett, they give us coupling, and coupling means you understand by how they becomes a pair, and repulsion means how they get segregated, and repulsion phenomenon. So this coupling and repulsion phenomenon, it is given by Bateson and Punnett, right? You understand? Second thing, linkage. This word is first used by this term. Term is given by, or we can say simply given by, is given by Morgan. Morgan, right? Actually, it is not GHUN, it is GAN. Hunt Morgan. He gave this term Morgan. Morgan, crossing over, sex-linked inheritance. These all terms was given by Morgan. So Morgan is also called as father of experimental biology. Experimental genetics, not biology, but genetics. Right? That Morgan. Morgan first discovered the sex linkage. Very first, and he used this term linkage, right? So what is the, we can say, what theory of linkage? According to the theory of linkage, some properties are there. Here we can say theory of linkage. So first means what are the criteria, what are the criteria or what are the properties of, simply properties of linkage. So first, that here genes are present in chromosomes, and these genes are present, linearly present, right? So we can say here the genes, linked genes, link means what? Linkage means what? It is the linked. That means genes, they are linked with chromosomes, right? So genes are linked chromosomes linearly, right? Here we can say genes are linked linearly on chromosome. First thing is here, so genes, this is genes, are linearly linked on chromosomes, right? And it get separate only through crossing over. First thing there, only through means only crossing over is the process in which genes they can exchange in homologous chromosomes, right? So all the genes they cannot exchange. It is depend upon the strength of linkage, right? The second thing was, now second point means, because I tell here strength of linkage, it is depend, crossing over means it is depend upon the strength of linkage that decide which genes means exchange or which are not exchange. Here we can say strength of linkage is in proportional to strength of linkage is in proportional to crossing over, or we can say distance also, right? Distance, more distance means more possibility of crossing over. Less distance means less possibility of crossing over. The strength of linkage is in proportional to, or we can say inversely proportional to This one is inversely proportional, right? The distance, or we can say inversely proportional to, that is crossing over. Right? Or, do you understand here? That means greater distance, greater possibility of crossing over. Less distance, less possibility of crossing over. Third point is here, what is that we say linkage group. Linkage group is what? It is the genes which are present in a haploid number of chromosomes. We can say here haploid number of chromosomes means what? The genes, haploid number of chromosomes are called the group, linkage group. As for example, we have 46 chromosomes. Haploid means what? 23 chromosomes. So the 23 chromosomes means these are called 23 groups of. How many groups are linkage groups are there? 23 linkage groups are there. Right here. That means third. Third means what? Linkage group. Linkage group means all the genes which are located on here. All the genes which are located on one pair of homologous chromosomes. Homologous chromosomes. As for example here, as for I tell you, humus means 46 chromosomes. That means a linkage group means 23 chromosomes. Another examples are also there. Suppose that a P means 14, 2N means 14 chromosomes. 2N, N means what? 7. That means linkage group is 7. Do you understand here? That means some example. P, 2N means 14, N means what happened? Haploid number means 7. That is the number of linkage group. Right? Second mage is there. Number of 2N. 2N means chromosome is 20. So haploid number means 10. That is the number of linkage group. Do you understand? Drosophila, which is used by Morgan. Drosophila means number of chromosomes is very less. In Drosophila, number of chromosomes is only 8. 2N means 8. That means what? N is 4 linkage group are there. Okay. Human is also there. So we can understand here that haploid number of chromosome is called linkage group. Linkage group. As for human means 46, linkage group is 23. Another examples are also there like barley. Barley means number of chromosomes is 14, same to P. 14, linkage group is 7. Rat is also there. Rat or mouse. Rat means how many number of chromosomes are there? Look here, humans means 46, but rat has 42 chromosomes. Linkage group means 21. Right? So this side N means what? It is linkage group. GAGE, linkage group, we can say. So do you understand? Now here, factors affecting the linkage group. Which type of factors are there who affect the linkage group? You can see here. Here, because who affect the linkage. Linkage means what? We understand the strength of linkage means what? It is depend upon the distance. More distance means strength is less. Right? So here we can say factors affecting the linkage group, or linkage, not group. We cannot say linkage group. Linkage simply, right? Linkage. Which factors are there? First, Here we can say distance, right? Distance. Distance means if distance increase means linkage crossing. Linkage means means crossing over. This crossing over is also increased. Possibility of crossing over is also increased, right? If distance increase, more distance means less strength, and that means what? Crossing over is increased. Second means what happen if temperature increase? Temperature increase. Temperature means simply in physics temperature increase means distance is also increase. So distance increase means what? Simply we know crossing over this means it is also increased, right? Third distance, temperature, now X-ray. X-ray, we give more X-ray means what? More chance of crossing over. Upper side means increase, right? Age, older age means less possibility of crossing over, right? Age increase, crossing over decrease, just opposite, right? And fifth one is, suppose that here means male or female. We can say here about the sex. In case of male means what? Decrease, crossing over possibilities decrease. As for example, in male Drosophila, crossing over is absent. So these are the factors which affect the linkage, right? Linkage means difference increase means linkage strength is decreased. Temperature increase means linkage strength is decreased. X-rays means linkage strength is decreased. But when age is more, linkage strength is increased. In male case, what happens? Because these linkages are very near, distance is very less. That means what happens here? No crossing over is there. No crossing over means there is no effect on linkage groups is there. Okay. Now you understand here something. Some two or three more points are there which I think we have to know. We need to know, right? And then after we start our NCERT. So some other means what happens here? We can say complete or incomplete or arrangement of link means genes. Arrangement of link, when we consider the arrangement of genes, that means what happens here? Two types of arrangement. One is called cis arrangement, another one is called trans arrangement. Cis and trans arrangement. Here we can say, and the term is arrangement of genes. Two types. Which two types? First one is called cis arrangement. Cis arrangement, and second one is called trans arrangement, right? So cis and trans arrangement. Cis arrangement means what? Cis means what? Here only means we can say here pure gametes are simply. Cis arrangement means when suppose that this one is chromosome, this one is another chromosome. They contains only dominant, capital A, capital T. They contains only recessive, capital A, small a. Here we can say A, B, this one is small a, small a. So this type of arrangement where both are dominant or both are recessive, then this one is called cis arrangement. This one is called cis arrangement. And trans arrangement means what? Trans means what? Capital A, small b, or capital B, small a. That is called trans. When they are transferred, right? So here Trans arrangement means what? Is due to the secretion. We can see here capital A, small b. Here small a, capital B. I can write here A, B, and here small a, capital B. This arrangement, this one we can see here. They are not means pure or they are not recessive or dominant. Means we can say it is hybrid, and this one is called trans arrangement. This one is trans arrangement. Simply, it is not a. We need not to go very deep. Only you have to understand means pure. Both dominants are present, and both means if both or all the recessives are present, that is called cis arrangement. Here, dominant and recessive both are present. Dominant, recessive both are present. That's why it is called trans arrangement. Do you understand? Trans arrangement means it is due to the crossing over. Due to the crossing over, trans arrangement happens. Because here what happens? These are transferred. B goes to this side and this B goes to that side. Right? That's one is called here. That's why it is called as trans arrangement. Because these genes are transferred. Simply they are transferred. Do you understand?